Okay, so welcome back to this next video in which we are discussing ion channels and cancer. Okay, right, so we're currently looking at different examples of ion channels. Okay, we've looked at potassium channels, we've now looked at voltage-gated sodium channels. There are just two more that I want to go over before we actually start looking at how ion channels are involved in the pathogenesis of cancer. Okay, so firstly I want to mention the voltage-gated calcium channels, and then we'll go on to the trip channels. Okay, so voltage-gated calcium channels, again, they have a major subunit which actually makes the pore-forming portion of the voltage-gated calcium channel, and then they have a lot of auxiliary subunits attached to them. Now we're only going to study the core pore-forming unit, okay, which in the case of voltage-gated calcium channels is called the alpha-1 subunit. So I'll just draw this here. So here is the core pore-forming unit, which actually has the pore down the middle, which calcium ions can move through, and this is now known as the alpha-1 subunit in voltage-gated calcium channels. And again, it has exactly the same membrane-spanning topology as the alpha subunits in voltage-gated sodium channels. It's a single polypeptide that has these 4 domain structures, where each domain has the same sort of structural features as a single subunit of a voltage-gated potassium channel. And effectively, you can think of it as 4 separate subunits of voltage-gated potassium channels stitched together to make one giant protein, which is capable of forming a pore-forming subunit on its own, basically. Okay, so in the case of voltage-gated calcium channels, there are 10 different alpha-1 subunits that you can use, okay? And these are grouped into 3 separate families. So there is the CAV1 family, there is the CAV2 family, and there is the CAV3 family. Now there are 4 members of the CAV1 family, which are called CAV1.1, CAV1.2, CAV1.3, all the way up to CAV1.4. Okay, there are 3 members of the CAV2 family, which are called CAV2.1, all the way up to CAV2.3. And then there are 3 members of the CAV3 family, which are called CAV3.1, all the way up to CAV3.3. Okay, so very simple naming system for the alpha-1 subunits of voltage-gated calcium channels. Okay, right, so those are, again, voltage-sensing channels. They will open in response to depolarization of the electrical potential difference across the cell membrane. And I'll stress again that, really, voltage-gated calcium channels, usually they have a lot more subunits than just the core pore-forming subunit. Here we are just looking at the single pore-forming unit, okay, but there are loads of accessory subunits that are usually attached onto the alpha-1 subunit to produce a normal voltage-gated calcium channel. Okay, right, so the final type of ion channel that I want to discuss is the family of trip channels, and we're going to see a few examples of these later on when we look at specific examples of ion channels that have been found to be involved in cancer. Okay, so TRIP stands for the transient receptor potential, okay? And you find transient receptor potential channels in a huge number of different cells, okay? So not just excitable cells. The transient receptor potential channels are, oops, I've missed off the potential word, transient receptor potential channel. They are famous for their role in sensory neurons, particularly their role in pain neurons, because many of them can respond to, for instance, noxious heat stimuli and noxious cold stimuli. Okay. However, you do find them in non-excitable cells as well as excitable cells. Okay, and they regulate the amount of sodium and calcium coming into the cell, because they are non-selective cation channels. So that's a key piece of terminology surrounding these channels. They are non-selective cation channels. So what this stands for is non-selective cation channel. Okay, right. let's have a look at the different members of the transient receptor potential channel family. So the subunits of transient receptor potential channels are divided into 7 major families. Okay, and let me tell you about these 7 major families now. So there is the trip A family. Okay, there's also the trip L family, the trip M family, the trip ML family, the trip N family, the trip P family, and then finally the trip V family. The examples that we're going to see later on are from the trip M family. Now, subunits of trip channels associate into tetramers, just like voltage-gated potassium channels, and indeed the subunit structure is again very similar to voltage-gated potassium channels. Okay, so here is the full channel sitting in the plasma membrane like so, and then it's made up of 4 separate subunits, which I'll draw now. So 1 there, second one there, third and fourth ones there. Okay, and if we take now one of these subunits out of the channel and have a look at its membrane spanning topology, what it looks like is like so. So you have, here's your first membrane spanning alpha helix, second, third, fourth, fifth P-loop, and then the 6th membrane spanning alpha helix like so, with both the amino and carboxy termini intracellularly. Okay, right, so that's the structure of trip channels. You take 4 trip subunits and put them together to make a full trip channel. And the examples that we're going to see are going to be homotetramers. And the subunits for trip channels are separated into these 7 different families. And the family that's going to be important for us is this trip M family. So we're going to look later at trip M1, trip M7, things like that. Okay, right. So that's the prerequisite portion on ion channels now finished. Let's now turn our attention to actually looking at how these things are going to be involved in cancer. So initially what I'm going to start off with is some general points about how these things might be involved in cancer, which go for absolutely all of the different ion channel families that we've now looked at. Okay? And then what I'm going to do is I'm then going to go on to looking at specific examples of specific ion channels that seem to have cropped up as being drivers in cancer. Okay, so the first thing then to say is that in cancer cells, often the ion channels that are expressed are completely abnormal, basically. Usually you have abnormal expressions of ion channels. So if you take a normal cell and a cancerous cell from the same tissue and compare which ion channels are expressed, generally it's very different, basically. So that's the first hint that ion channels might have a role in cancer. Okay, then the next thing that I want to talk about is the fact that cancer cells, and this is a phenomenon that you find all over the place, okay, general phenomenon, cancer cells generally have a depolarized membrane potential. Okay, so the electrical potential difference across the cell membrane is generally depolarized in cancer cells. Okay, so if you look at this electrical potential difference, it's generally between negative 20 millivolts and negative 40 millivolts, so somewhere around there. So usually resting membrane potential, as we said before, is around negative 60 millivolts. So this is quite depolarized, basically. Okay, now, what we wanted to see, well, what people wanted to see is this important? Is this important for the cancerous phenotype? And it seems as though it is very important for the cancerous phenotype. Okay, because if you actually take a non-cancerous cell, and I believe this experiment was done actually in a completely terminally differentiated cell. So if you take a neuron, okay, and neurons do not divide. They are what are known as terminally differentiated cells. I'll put this here. They are not usually capable of dividing. The general principle is that there is no cell division in the brain. It's not quite true. There is a bit in the hippocampus, okay? But the general principle is that the cells that you have in your brain now are the same cells as you had when you were born, basically, okay? So neurons are not dividing. It's a terminally differentiated cell. Okay, right, so you take this neuron, and what you do is you expose it to sustained depolarization of its electrical potential difference across its cell membrane. So through voltage clamp apparatus, we can set the electrical potential difference across cells. So basically we can take the cell out and we can set up apparatus so that we can control the electrical potential difference across the cell membrane of the neuron. So I just draw a little picture of the neuron here. Those are the dendrites there. Here's the axon with the axon terminal. Okay, we are now going to set the electrical potential difference across that cell membrane of the neuron. And basically, we're going to set it up to a depolarized level, and we're going to sustain that for a long period of time. Okay, so let's say we sustain it at, let's say, for a good average, we'll sustain it at negative 30 millivolts. Well, what they have actually found is that if you sustain a depolarization across the cell membrane of a neuron, you can actually get this neuron to start doing processes that are similar to division. You can't actually get it to divide. I want to stress that. We have not been able to get it to divide yet, but it starts doing processes that are involved in division, odd processes that are not normal for neurons. Okay, so it starts DNA synthesis, it starts replicating its DNA, okay, and also it starts assembling mitotic spindles which are involved in mitosis, okay, and are important for splitting sister chromatids apart, but which normal cells do not have. They usually do not have their microtubules arranged in mitotic spindles. They do have microtubules, but they're involved in the normal cytoskeleton. So mitotic spindles are an abnormal portion of the cytoskeleton, basically, an abnormal cytoskeletal structure to have within a neuron. Okay, so you start getting the assembly of mitotic spindles and DNA synthesis. These are both activities associated with division. So although we can't get the neuron to actually divide, by sustaining depolarization you can get it to show behavior which is involved in division, and maybe that shows us that the sustained depolarization is important for cancer cells being able to divide rapidly. Okay, right, this goes further. Okay, so we've taken a normal cell, exposed it to sustained depolarization, and seen maybe that it's now starting to show behavior associated with division. What's the other thing that we could do? Take a cancerous cell and change its electrical potential difference back to the normal electrical potential difference and see if that stops it dividing, and it does. Okay, if you get rid of the sustained depolarization, the cancer cell stops dividing, it arrests division. Okay, so this is interesting. Okay, what determines the electrical potential difference across cell membranes? Well, it's ion channels. Okay, so this basically blatantly says ion channels seem to have some essential role in cancer, but maybe it's just an essential role in division. It might not be necessarily that they are the drivers in the carcinogenic phenotype. Okay, but this is the first major link between ion channels and cancer, that cancer cells have this chronic, sustained, depolarized electrical potential difference across their cell membranes, and that this seems to be essential for them being able to divide again and again and again very, very rapidly. And indeed, if you do it to a normal cell that you would usually never be able to get to divide, it can actually begin division-like behavior. Okay, so the major ion channels that are involved in determining resting membrane potential are those twin-pore potassium channels, twin-pore domain potassium channels. I made the horrendous mistake that I warned you against. Okay, so the K2P channels. Okay, so maybe their dysregulation is what leads to the sustained depolarization across the membrane of the cancerous cells. Okay, right, so that's a first general point that relates to not just a specific type of voltage-gated potassium, not just a specific type of ion channel, but potentially to the full complement of ion channels. And I want to just say something here that's important. Okay, when you look at cancer cells, okay, we've said that they all have dysregulated ion channel expression, but if you take cancer cells from two different tumours, okay, so we'll have cancer cell from tumour number 1 here, and cancer cells from tumour number 2 here. basically, if you look at the expression of ion channels in cancer cell number 1 and compare it to the ion channels in cancer cell number 2, they will not be the same, basically. Okay, so it's not as though there is some change that is the same for all different cancer cells in all different tumours, basically. The dysregulation, the way that ion channels become dysregulated in their expression in cancer cells is different for cancer cells from all different tumours, basically. Okay, so it's not as though there is one change that then causes the depolarization and therefore causes the overproliferation. Okay, it seems as though it's different ion channels becoming dysregulated in different cancer cells that then causes the same overall phenotype, which is this eventual depolarization. So basically it's not just one ion channel that has changed in all cancer cells to cause this sustained depolarization. It's different between different cancer cells. Okay, so the question then becomes, is there some master regulator that regulates the expression of all potassium channels, okay, and which has been dysregulated in cancer? And that's an open question, we don't know yet. Okay, right, so that's one general point that I want to make. The other general point linking ion channels to cancer is the role of fluctuations in the electrical potential difference across the cell membrane in the progression of the cell cycle. So let me just tell you a little bit about the cell cycle then. Okay, so the cell cycle then is the process by which cells can divide into two. Okay, so it's split into 4 different phases. Okay, so the first phase I'll put here, which is known as the first growth phase, so G1. Okay, and in fact, before G1 I should probably put in another phase here, which is G0. So G0 is the phase in which a cell is not actually actively dividing. Okay, so when a cell goes from being in this phase G0 to being in the active cell cycle G1, we call that the G0, G1 transition, and all sorts of growth factors will stimulate a cell to move from the G0 phase to being in the G1 phase. Okay, so let's colour in these different portions. So we'll have G1 here in blue, and we'll have G0 here in orange. Okay, so after G1 then, oh actually I haven't told you what happens in G1. In G1 what happens is the cell begins to grow, okay, and also prepares for copying the DNA. Okay, so if we think about what needs to happen then for a cell to go from being one cell to being 2 cells, okay, so what needs to happen in cell division? Basically, the cell needs to grow in order to split into 2 cells, because it's going to have to duplicate all of its proteins. It's going to have to go from having enough proteins for 1 cell to having enough proteins for 2 cells. Okay, this is one of the things that's going to be happening in this first growth phase. So this is known as the first growth phase. Okay, basically what's going to be happening is the cell is going to be making more proteins so that it's got enough proteins for 2 cells rather than just 1 cell. Okay, right. In addition, it's going to be getting ready for the next portion of the cell cycle. So let me tell you about the next portion of the cell cycle. So the next portion of the cell cycle is what's known as the S phase, which stands for the synthesis phase. So this is synthesis here. And this is the phase of the cell cycle where you copy all of the DNA in the nucleus of the cell. Okay, so all 46 chromosomes in the case of human cells are going to be copied in the synthesis phase, and you're going to get exact replicas of those chromosomes known as sister chromatids. Okay, so basically the machinery for copying all of the DNA is not normally activated, and some of it's not even present in a normal undividing cell. So in the G1 phase you're going to get all of that machinery assembled and produced, basically, so that you're ready to begin the synthesis phase when that commences. Okay, so in G1 phase you're making more proteins and also preparing the machinery for copying the DNA. In the S phase then, what's going to happen is you're going to alter the nucleus now. So let's have a look at the nucleus. So in the nucleus you'll have 46 chromosomes. I'm only showing one chromosome here because I don't want to have to draw out all 46. And by the end of S phase, you're going to have replicated every single one of those chromosomes. So again, I'll just show this like so. There is this chromosome's sister chromatid. Okay, it's exact replica copy now. Okay, then after S phase, what you're going to have is a phase known as the second growth phase, which I'll put here. So this is known as G2, and this is the second growth phase. And in the second growth phase, what's going to happen is the cell is going to make more proteins again. So the noble work of the first growth phase is going to continue in the second growth phase. You're going to start making more proteins, okay, and continue this duplication of proteins that is needed in order for the cell to divide. In addition, you're going to be preparing for the final phase of the cell cycle, which is the M phase, okay? So here then, this is the M phase. And in the M phase, which stands for the mitosis phase, this is the phase where firstly you get the nuclear division, and then you get the cellular division. Okay, so in M phase, what happens is firstly you go from having a single nucleus, which has double the amount of DNA it needs, 2 copies of every single chromosome, to having a cell with 2 nuclei. Okay, so let me just show this. A cell with 2 nuclei, and each of these nuclei contains a single copy of all 46 chromosomes. And then finally, at the end of M phase, a process known as cytokinesis occurs. And in cytokinesis, you go from having a single cell with 2 nuclei in to having 2 cells, each with a single nuclei in, or rather a single nucleus in. Okay, so M phase, although it is called the mitosis phase, it's actually split into 2 separate portions, which is the mitosis portion, which means nuclear division. Mitosis literally means the division of a single nucleus, which has a replica of every single one of the 46 chromosomes, to having 2 separate nuclei which are identical. Okay, and then the final process is then cytokinesis. So M phase contains both mitosis and cytokinesis. So going back to G2 phase then, in G2 phase you're not just going to be duplicating proteins so that the cell is going to have enough proteins for 2 cells when it divides, but you're also going to be getting ready for M phase. You're going to be getting the machinery that's needed to split the nucleus apart ready, and you're also going to get the machinery that's needed for splitting the cell apart, so the machinery for cytokinesis. Okay, so these are the different phases of the cell cycle. Now, there are several very, very important checkpoints in the cell cycle, and the really important ones here are this first checkpoint here. Okay, that's a difficult one to get past. Going from G0 phase, or indeed from the M phase into the G1 phase. So the reason the cell cycle is called a cycle is because some cells can go straight back into G1 phase after having gone through M phase. So after the cell has divided into 2, these ones can go straight back into the division process, going into G1 phase and then go round again, and each of those will then divide into 2 cells. Okay, but this is one of the key checkpoints that you have to cross, either between G0 and G1 or between M and G1. That's a difficult point to cross. Okay. In addition, another key checkpoint is the checkpoint between G1 and S. That's a major checkpoint because that really represents where there's no turning back, basically. Once you have copied all of your 46 chromosomes, that really commits you to division then. Okay, so that's again a very important checkpoint. And then finally, another important checkpoint is the G2M checkpoint, which is where you begin this very dangerous process of actually splitting the nucleus apart and splitting the cell apart. That requires incredibly well orchestrated cell processes, okay? And if that goes wrong, it can result in terrible abnormalities in the daughter cells. Okay, so again, that's a very important checkpoint. So this is the G0 to G1 transition. This is the G1 to S transition. and this is the G2 to M transition, and those are very important transitions which have strict checkpoints. Now, at these three important checkpoints, what you find is fluctuations in the electrical potential difference across the cell membrane occurring, okay? And this is believed, again, to involve ion channels. Well, it has to involve ion channels. They control the electrical potential difference across the cell membrane. Okay, so let's start with this one between G1 and S. Actually, maybe it makes more sense to start with the one between G0 and G1. Okay, so the G0, G1 transition is marked by a depolarization. Okay, so when cells move from being in the G0 phase of this cell cycle, which is an inactive phase of the cell cycle, to being in the G1 phase, which is an active phase of the cell cycle, that is marked by a depolarization, a transient depolarization. It doesn't last. It's just a very quick transient depolarization of the electrical potential difference. Okay, so if we draw this, let's say this is our cancer cell here, okay? It's already got its highly depolarized electrical potential difference. When it goes through the division cycle, it will have a transient depolarization like that, further depolarization at the G0 to G1 transition, okay? Then between the G1 and S transition, okay, you get hyperpolarization, the opposite change in the electrical potential difference. Okay, so drawing that on my graph again, that will be a deflection downwards, okay, and again it's a transient hyperpolarization, okay, and then finally, at the G2 to M transition, you again get depolarization, a transient depolarization, and these changes in the electrical potential difference are believed to actually be very important in moving between one phase of the cell cycle and the other phase of the cell cycle, and indeed they may be a way of stopping the progression through the cell cycle by stopping these electrical potential difference changes. Okay, so let's have a look at these in a little bit more detail then. This hyperpolarization that is caused here at the transition between G1 and S, it is absolutely known that this is caused by potassium channels opening and allowing potassium out of the cell, which then hyperpolarizes the electrical potential difference across the cell membrane. Okay, so there's an involvement of potassium channels. Okay, and in the G2M transition, the thing that causes the depolarization here is believed to be chloride channels opening. Okay, and we haven't discussed chloride channels in detail, we haven't looked at their structures in detail, but we will be seeing several examples of chloride channels throughout this video. Okay, so chloride channels opening at the G2 to M transition allow chloride anions to leave the cytoplasm of the cell, and that then leads to the depolarization at the G2 to M transition. Okay, so again, here is something where ion channels are extremely important in the cell division cycle, which is of course extremely important in the pathogenesis of cancer, since the leading phenotype of cancer is the overproliferation. Okay, so potentially aberrant expression of ion channels could be involved in guiding cells more rapidly through the cell cycle. Okay, and that's potentially something that might develop in the future. Okay, so there's another general point linking ion channels to a phenotype of cancer. Okay, right. In the next video, what we're going to begin is our study of individual cases, okay? And we're going to start off by looking at Eferagogo-1 and Eferagogo-2. And Eferagogo-1 in particular is a very hopeful target for cancer in the future.